Hello everyone, welcome to NCERT Audiobook. Today we start the class 12th, lesson 5th, Principles of Inheritance and Variations. Have you ever wondered why an elephant always gives birth only to the baby elephant and not some other animal? Or why a mango seed forms only a mango plant and not any other plant? Given that they do, are the offspring identical to their parents or do they show differences in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other or sometimes even so different? These and several relation questions are dealt with scientifically in a branch of biology known as genetics. This subject deals with the inheritance as well as the variations of the characters from the parents to the offspring. Inheritance in the process by which the characters are passed on from parent to the progeny, it is the basis of heredity. Variation is the degree by which the progeny differ from their parents. Humans knew from as early as 18,000 to 1000 back that one of the causes of variation which hidden in sexual reproduction, they explored the variations that were naturally present in the wild populations of plants and animals to selectively breed and select for organisms that possessed desirable characters. For example, through artificial selection and domestications from the ancestor wild cow, we have well-known Indian breed. Example, Sahiwal cow in Punjab. We must, however, recognize that true, our ancestors knew about the inheritance of the characters and variations. They had very little idea about the scientific basis of these phenomena. Mendel's law of inheritance. It was during the mid 19th century that headway was made in the understanding of inheritance. George Mendel conducted the hybridization experiment on the garden pea for 7 years from 1856 to 1863 and proposed the law of inheritance in living organisms. During the Mendel's investigations into inheritance pattern, it was for the first time that statistical analysis and the mathematical logic were applied to problem in biology. His experiments had a large sampling size which gave together greater credibility to the data that he collected. Also, he confirmations of his inferences from the experiments on successive generations of his test plants prove that his result pointed to the general rules of inheritance rather than being understandable ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposite states. Example, tall or dwarf plant, yellow or green seeds. This allow him to set up a basic framework of rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observations and the complexity inheritance in them. Mendel conducted such artificial pollination, cross-pollination experiments using several true breeding pea lines. A true breeding line is one that, having undergone continuous self-pollination, show the suitable trait inheritance and expression for several generations. Mendel selected 14 true breeding pea plant varieties as pair which were similar except for one character with contrasting trait. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated, full or constricted pod, green and yellow pods, and tall and dwarf plants. Inheritance of one gene. Let us take an example of one such hybridization experiment carried out by the Mendel, where he crossed the tall and dwarf pea plant to study the inheritance of one gene. He collected the seed produced as a result of these cross and grew them to generate plants of the first hybrid generation. This generation is also called as a filial progeny or F1. Mendel observed that all the F1 progeny plants were tall, like one of its parents. None were dwarf. He made similar observations for the other pairs of trait. He found that the F1 always resembled either one of the parents and that trait of the other parent was not seen in them. Mendel then self-pollinated the tall F1 plants and, to his surprise, found that in the filial 2 generation, some of the offspring were dwarf. The characters was not seen in the F1 generation was not expressed. This proportion of the plants that were dwarf were 1/4 of the F2 plants, while 3/4 of the F2 plants were tall. The tall and dwarf trait were identical to their parental type and did not show any blending. That is, all the offsprings were either tall or dwarf. None of were of in between the height. Similar results were obtained with the other trait that he studied. Only one of the parental trait was expressed in the F1 generation, while at F2 stage, both the traits were expressed in the proportion of 3:1, and the contrasting trait did not show any blending at either F1 or F2 stage. Based on these observations, Mendel proposed that something was being stable passed down unchanged from parent to offspring through the gametes every successive generation. He called these things as a factor. Now we call them as a genes. Genes, therefore, are the unit of inheritance. They contain the information that is required to express the particular trait in an organism. Genes which code for a pair of contrasting traits are known as allele. That is, they are slightly different from one of the same gene. If we use the alphabetical symbols for each gene, then the capital letters is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of the characters of height, capital T is used for the tall trait and small t is used for the dwarf. And the capital T and small t are alleles of each other. Hence, the plants, the pair of the allele for height would be capital T, capital T small t or small t small t. Mendel also proposed that in a true breeding, tall or dwarf P variety, the allelic pair of the genes for height are identical or homozygous, capital T and small t respectively. Capital T and small t are called genotype of the plant, while the description terms of the tall and dwarf are the phenotype. What then would be the phenotype of the plant that had a genotype capital T small t? As Mendel found the phenotype of the F1 heterozygous capital T small t to be exactly like of the small capital T parents in appearance, he proposed that in a pair of dissimilar factor, one dominance of the other, and hence they are called as the dominant factor while the other called as a recessive factor. In this case, capital T is the dominant over the small t or recessiveness. That is the recessive. He observed the identical behavior for all the other characters, trait pairs that studied. It is convenient to use the capital and lowercase of the alphabetical symbol to remember this concept of dominance and recessiveness. Do not use capital T for tall and D for the dwarf because you will find it difficult to remember when the capital T and D are the alleles of the same gene or character or not. Alleles can be similar as in the case of homozygous capital T and small t or can be dissimilar is in the case of heterozygous capital T small t. Since the capital T plant is heterozygous for gene controlling one character, height, it is the monohybrid, and the cross between capital T and small t is the monohybrid cross. From the observation that the recessive parents trait is expressed without any blending in F2 generation, we can infer that when the tall and dwarf plant produce gametes by the process of meiosis, the allele of the parental pair separates or segregate from each other and only one allele is transmitted to the gamete. This segregation of the allele is a random process and so there is a 50% chances of the gamete containing either allele as has been verified by the result of crossing. In this way, the gametes of the tall T plants have the allele capital T and the gametes of the dwarf small t plants have the allele small t. During fertilization, the two allele capital T from the one parent say through the pollen and the small t from the other parent then through the egg are united to produce zygote that have one capital T allele and one small t allele. In other words, the hybrid have capital T small t. Since these hybrids contains allele which express contrasting trait, the plants are heterozygous. The production of gametes by the parents, the formation of zygote and the F1 and F2 plants can be understood from the diagram called Punnett square. It was developed by the British scientist Reginald C. Punnett. It is the graphical representation to calculate the probability of the all possible genotypes of offspring in the genetic cross. The possible gametes are written on two sides, usually the top row and the left column. All possible combinations are represented in boxes below the squares, which generates a square output form. The Punnett square has shown in the parental tall T male and dwarf T female. The gametes produced by them and the F1 T small t progeny, the F1 plants of the genotype T small t are self-pollinated. The symbols of female and male are used to denote the female egg and male pollen of the F1 generation, respectively. The F1 parent of the genotype T small t, when self-pollinated, produce the gametes of the genotype T and small t. Genotype T have a 50% of the chance to pollinate eggs of the genotype T as well as the genotype small t also pollen grains to genotype small t have a 50% chance to pollinating eggs to genotype T as well as of genotype small t. As a result of random fertilization, the resultant zygote can be of the genotypes T small t or small t. From the Punnett square, it is easily seen that 1/4 of the random fertilizations lead to T, half leads to T small t and 1/4 leads to small t. Through the F1 have the genotype of T small t but the phenotypic characters seen in tall at the F2 3/4 of the plants are tall, where some of them are T while others are the T small t. Extremely, it is not possible to distinguish between the plants with the genotype T and T small t. Hence, within the genotypic pair T small t, only one character T tall is expressed. Hence, the character T or tall is said to be dominant over the other allele small t or dwarf character. It is thus due to this dominance of one character over the other that all the F1 are tall through the genotype T and small t, and in the F2 3/4 of the plant are tall through the genotypically of 1/2 T small t and only 1/4 are the T. This leads to a phenotypic ratio 3/4 tall, 1/4 T, 1/4, 1/2 T small t and 1/4 of the small t, that is the ratio 3:1 ratio. But the genotypic ratio is 1:2:1. The 1/4 and 1/2 and 1/4 ratio of T small t and small t is mathematically considerable to form the binomial expression ax plus by raised to power 2 that has the gamete bearing genes capital T and small t in equal frequency of half, and the expression is expanded in given below. Mendel self-pollinated the F1 plants and found the dwarf F2 plants continued to generate the dwarf plants in F3 and F4 generation. He concluded that the genotype of the dwarf was homozygous small t. What do you think he would have got had he self-pollinated a tall F2 plant? From the preceding paragraphs, it is clear that through the genotype ratio can be calculated using mathematical probability by simply looking at the phenotype of the dominant trait. It is not possible to know the genotype composition, that is, for example, whether a tall plant from a F1 and F2 has capital T, small t composition cannot be predicted. Therefore, to determine the genotype of tall plants at F2, Mendel's crossed the tall plant from F2 with the dwarf plant. This he called a test cross. In a typical test cross, an organism plant here showing a dominant phenotype and whose genotype is to be determined. He crossed with the recessive parent instead of the self-crossing. The progenies in such a cross can easily be analyzed to predict the genotype of the test organism. The results of the typical test cross where the violet color flowers is dominant over the white color flower. Using Punnett square to try find out the nature of offspring of the test cross. What ratio did you get? Using the genotype of this cross, can you give a general definition for the test cross? Based on his observation on monohybrid crosses, Mendel proposed two general rules to consolidate these understanding and inheritance in the monohybrid crosses. Today these rules are called the principles or laws of inheritance. The first law of the law of dominance and the second law is law of segregation. Law of dominance. Characters are controlled by discrete units called factors. Factors occur in pairs. In dissimilar pair of the factors, one membrane of the pair dominates is called dominant and other is recessive. The law of dominance is used to explain the expression of only one of the parental character in a monohybrid cross in the F1 and expression of both in F2. It also explains in the proportion of 3:1 obtained at the F2. This law is based on the fact that its allele do not show any blending and that both the characters are recovered as such in the F2 generation through one of these is not seen at the F1 stage. Through the parent contain two alleles during gamete formation, the factors or the alleles of the pair segregates from each other such that the gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of the gametes, each having one allele with the equal proportion. Incomplete dominance. When experiments on peas were repeated using other trait in other plant, it was found that sometimes the F1 had phenotype that did not resemble either of the two parents and was in between the two. The inheritance of the flower color in the dog flower, snapdragon and the anthridium species is a good example to understand the incomplete dominance. In a cross between the true breeding red flower and true breeding white flower, the F1 was pink. When the F1 was self-pollinated, the F2 resulted the following ratio: 1 red, 2 pink and 1 white. Here the genotype ratio were exactly as we would accept in any Mendelian monohybrid cross, but the phenotypic ratio is changed from 3:1 dominant recessive ratio. When happened was that R is not completely dominant over the small r, and this made the possible to distinguish capital R as a pink from the capital R red and small r white. Explanation of the concept of the dominance. What exactly is dominance? Why are some allele dominant and some are recessive? To tackle this question, we must understand what a gene does. Every gene, as you know by now, contains the information to express the particular trait in a diploid organism. There are two copies of each gene, that is, as pair of allele. Now, these two alleles need not always be identical, as in a heterozygote one of them may be different due to some changes that it has undergo, about which you will read further on and in the next chapter, which modifies the information that a particular allele contains. Let's make the example of gene that contains the information for producing the enzymes. Now, there are two copies of these genes, the two allelic forms. Let us assume that the normal allele produces the normal enzyme that is needed for the transformation of the substrate S. Theoretically, the modified allele could be responsible for the production of the normal less efficient enzyme or a non-functional enzyme or non-enzyme at all. In first case, the modified allele is the equivalent to the unmodified allele. That is, it will be produces the same phenotype trait that is the result of the transformation of substrate S. Such equivalent allele pairs are very common, but if the allele produces non-functional enzyme or no enzymes, the phenotype may be affected. The phenotype trait will be dominant dependent on the functioning of the unmodified allele. The unmodified functioning allele, which represent the original phenotype, is the dominant allele, and the modified allele is generally is the recessive allele. Hence the example above, the recessive trait is seen due to the non-functional enzymes and because no enzymes is produced. Codominance. Till now we were discussing crosses where the F1 resembles either of the two parents, dominance, or was in between, incomplete dominance. But in case of codominance, the F1 generation resembles both the parents. A good example is the different types of the red blood cells that determine ABO blood grouping in human being. ABO blood groups are controlled by the gene I. The plasma membrane of the red blood cells has sugar polymer that produced from its surface, and the kind of the sugar is controlled by the gene. The gene I is the three allele IA, IB and small I. The alleles IA and IB produce the slightly different forms of the sugar, while the allele small I does not produce any sugar. Because humans are diploid organisms, each person possesses any two or three I gene allele. IA and IB are completely dominant over the small I. In other words, when IA and small I are present, only IA express, and when the IB and small I are present, IB express. But when the IA and IB are present together, they both express their own types of sugar. This is because of the codominance. Hence, red blood cells have both A and B types of sugar. Since there are many different alleles, there are 6 different combinations of these 3 alleles that are possible, and therefore the total 6 different genotypes of the human ABO blood types. How many phenotypes are possible? Do you realize that the example of ABO blood grouping is also provides a good example of multiple alleles? Can you see that there are more than two, that is three alleles governing the same character? Since the individual only two alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene produced may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene and has two alleles. Starch is synthesized effectively by B and homozygous, where therefore the large starch grain are produced. In contrast, the small b homozygous have lesser efficiency in starch synthesis and produces smaller starch grain. After maturation of these seeds, B seeds are round and small b seeds are wrinkled. Heterozygous produce the round seed and the so B seems to be the dominant allele. But the starch grains produced are the intermediate size of the capital B small b seeds. So if the starch grain size is considered as a phenotype, then from this angle the alleles show incomplete dominance. Therefore, dominance is not a autonomous features of the gene or the product that is has information for. It depends as such much on the gene product and production of the particular phenotype from this product as it does on a particular phenotype that we choose to examine in case more than the one phenotype and influenced by the same gene. Inheritance of two genes. Mendel also worked with the crossed pea plant that different in two characters as is seen in the cross between the pea plant that has seeds with yellow color and round shape and one that had seeds of green color and wrinkle shape. Mendel found that the seeds resulting from the crossing of the parents had yellow colored and round shaped seed. Here, can you tell which of the character in the grain pair's yellow, green color and round wrinkle shape was dominant? Thus, yellow color was dominant over the green and round shape dominant over the wrinkle. These results were identical to those that he got when he made separated monohybrid crosses between the yellow and green seeded plants and between the round and wrinkle seeded plants. Let us use the genotypic symbol capital Y for dominant yellow seed color and small y for the recessive green seed color. Capital R for the round shape seed and small r for the wrinkle seed shape. The genotype of the parents can be then be written as the capital R, capital Y and small r, small y, small y. The cross between the two plants can be written down as in figure, showing the genotype of the parent plant. The gametes capital R, capital Y and small y unite to fertilize to produce the F1 hybrid capital R, capital Y, small y. When the Mendel self-hybridized the F1 plants, he found that 3/4 of the F2 plants had yellow seeds and 1/4 had green. The yellow and green color segregate in the ratio of 3:1. Round and wrinkle seed shape also segregated in the form of 3:1 ratio, just like a monohybrid cross. Laws of independent assortment. In the dihybrid cross, the phenotype round yellow wrinkled, yellow round green and wrinkled green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of the characters that Mendel studied. The ratio 9:3:3:1 can be derived as the combination series of the 3 yellow, 1 green with 3 round and 1 wrinkled. This derivation can be written as follows: 3 round, 1 wrinkled, 3 yellow, 1 green, 9 round yellow and 3 wrinkled yellow, 3 round green and 1 wrinkled green. Based upon such observation on the dihybrid crosses, Mendel proposed the second set of the generations that we call the Mendel's law of independent assortment. The law states that when two pairs of the trait are combined in a hybrid, segregation of one pair of the character is independent of the other pair of the characters. The Punnett square can be effectively used to understand the independent segregation of the two pairs of the genes during meiosis and the production of egg and pollen in the F1 RRYY plant. Consider the segregation of the one pair of the gene capital R and small r. 50% of the gametes have the gene capital R and the other 50% have a small r. Now besides each gamete having either capital or small r, it should also have the allele capital Y or small y. The important thing to remember here is that segregation of the 50% are important thing and 50% of the small r is the independent form of the segregated and 50% of the capital Y and 50% of the small y. Therefore, 50% of the small r bearing the gametes and bearing gametes Y and the 50% of the small y. Thus, there are four genotypes and gametes. The four types are capital R, capital R, small r, capital Y, and small r, small y, each with a frequency of 25% or one-fourth of the total gamete produced. When you write down the four types of the eggs and pollen on the two sides of the Punnett square, it is very easy to derive the composition of the zygote that gives rise to the F2 plants. Although there are 16 squares, how many different types of the genotypes and phenotypes are formed? Note them down in the format given. Can you, using the Punnett square data, work out the genotypic ratio at the F2 stage and fill in the format given? It is the genotypic ratio, also 9:3:3:1. Chromosomal theory of inheritance. Mendel published his work on the inheritance of characters in 1865, but for the several reasons it remained unorganized, unrecognized till 1900. Firstly, communication was not easy. In those days, his work could not be widely published. Secondly, his concept of the gene or factor in the Mendel's word as stable and discrete units that control the expression of the trait and of the pair of the allele, which did not blend with each other, was not accepted by his contemporaries as in the explanation for the apparently continuously variations seen in the nature. Thirdly, Mendel's approach of using the mathematician to explain the biological phenomena was totally new and unacceptable to many of the biologists in this time. Finally, through the Mendel's work suggested that the factors were discrete units, he could not provide any physical proof for the existence of the factors or say what they were made of. In 1900, three scientists, De Vries, Korean, and the von Teschmark, independently discovered the Mendel's result on the inheritance of characters. Also by this time, due to advancement of the microscopy and were taking place, scientists were able to carefully observe the cell division. This led to the discovery of the structure in the nucleus that appeared to double and divide just before each cell division. These were called the chromosomes. By 1902, the chromosomes movement during meiosis had been worked out. Walter Sutton and Theodore Boveri noticed that the behavior of the chromosome was parallel to the behavior of the gene and used to the chromosome movement to explain the Mendel's law. Recall that you have studied two behavioral chromosomes during mitosis and during meiosis. The important thing to remember are that chromosomes are as well as genes occur in pairs. The two alleles of the gene pair are located on homologous sides of the homologous chromosomes. During anaphase of meiosis 1, the two chromosome pairs can align at the metaphase plate independently of each other. To understand this, compare the chromosomes for four different colors in the left and right columns. In the left column, orange and the green of segregating together, but in the right column, possibilities of the orange chromosome in segregating with the red chromosomes. Sutton and Boveri argued that the pairing of the separations of the pair of chromosomes would lead to the segregations of a pair of factors they carried. Sutton united knowledge of the chromosomal segregation with the Mendelian principles and called it the chromosomal theory of the inheritance. Following this synthesis of ideas, experimental verifications of the chromosomal theory of inheritance by Thomas Hart Morgan and his colleagues led to discovering the basis for the variations of sexual reproduction produced. Morgan worked with the tiny fruit flies Drosophila melanogaster, which were found very suitable for such study. They could be grown to simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks and the single mating could produce a large number of the progeny flies. Also, there was a clear differentiation of the sexes. The male and female flies are easily distinguishable. Also, it has many types of the hereditary variations that can be seen with low power microscope. Linkage and recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to the dihybrid crosses carried out by the Mendel in P. For example, Morgan hybridized yellow-bodied, white-eyed females to the brown-bodied, red-eyed males and intercrossed with their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio derived very significantly from the 9s to 3s to 3s to 1 ratio. Morgan and his group knew that the genes were located on the X chromosomes. Dihybrid cross were situated on the same chromosomes. The production of the parental gene combined were much higher than the non-parental type. Morgan attributed this due to the physical association of the linkage of the two genes and coined the term linkage to describe this physical association of the genes on the chromosomes and the term recombinations. To describe the generation of the non-parental gene combinations, Morgan and his group also found that even when the genes were grouped on the same chromosome, some genes were very tightly linked while others are loosely linked. For example, he found that genes white and yellow are very tightly linked and showed only with 1.3% of the recombinations, while white and miniature wing showed 37.2% recombination. He studied Alfred Stutterwant used the frequency of the recombination between the gene pair on the same chromosomes as the measure of the distance between the genes and mapped their positions on the chromosomes. Today, genetic maps are extensively used as a starting point of the sequencing of whole genomes, as was done in case of the human genome sequencing project described later. Sex determination. The mechanism of the sex determination has always been puzzled before the genetics. The initial clue about the genetics chromosomal mechanisms of the sex determination can be tracked back to some of the experiments carried out by the insects. In fact, the cytological observation made in the number of the insect lead to development of the concept of the genetic chromosomal basis of the sex determination. Hanking 1891 could trace the specific number nuclear structure, although the spermatogenesis is a few insect, and it was also observed by him that 50% of the sperm received the structure after spermatogenesis, whereas the other 50% sperm did not receive it. Hanking gave the name of this structure as a X body, but he could not explain its significance. Further, the investigation by other scientists led to the conclusion that X body of Hanking was in fact of the chromosome. That is why it was given the name X chromosome. It was also observed that in a large number of the insects, the mechanism of the sex determination is of the XO type. That is, all eggs bear the additional X chromosome besides the other chromosomes or autosomes. On the other hand, some of the sperms bear the X chromosome, whereas some of do not. Eggs fertilized by the sperm having an X chromosome become the females, and those fertilized by the sperm that do not have a X chromosome become males. Do you think the number of the chromosome in the male and females are equal? Due to the involvement of the X chromosome in the determination of the sex, it was designed to be sex chromosome, and the rest of the chromosome were named as autosomes. Grasshopper in the example of XO type of the sex determination, in which the males have only one X chromosome beside the autosomes, whereas female has a pair of X chromosome. These observations lead to the investigations of the number of the species to understand the mechanism of the sex determination. In the number of other insect and mammals, including man, XY type of the sex determination is seen, where both the male and female have the same number of the chromosomes. Among the males, an X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. Females, however, have a pair of X chromosomes. Both males and females bear same number of autosomes. Hence, the males have autosomes plus XY while the females have autosomes plus XX. In human beings, the Drosophila and the males have one of the X and one Y chromosome, whereas female have a pair of X chromosomes beside the autosomes. In the above description, you have studied about two types of the sex determining mechanisms, that is XO type and XY type, but in both cases, male produce two different types of the gametes, either with or without X chromosome, or be some gametes with X chromosomes and some with Y chromosomes. Such types of the sex determination mechanism is designed to be the example of male heterogamety. In some other organisms, birds, and different mechanism of the sex determination is observed. In this case of the total number of the chromosome is same in both males and females, but two different types of the gametes is term for the sex chromosome are produced by females, that is female heterogamety. In order to have a distinction with the mechanism of the sex determination described earlier, the two different sex chromosomes of the female bird has been designed to be the Z and W chromosomes. In these organisms, the females have a one Z and one W chromosome, whereas the male have a pair of Z chromosomes beside the autosomes. Sex determination in human. It has already been mentioned that the sex determining mechanism in the case of human is XY type. Out of 23 pairs of the chromosome present, 22 pairs are exactly same in both males and females. These are the autosomes. A pair of X chromosomes are present in the females, where the pair of X and Y chromosomes are determined the male characteristics. During spermatogenesis, among the male, two types of the gametes are produced. 50% of the total sperm produced carry X chromosome, and the 50% of the total sperm carry the Y chromosome besides the autosome. Females, however, produce only one type of the ovum with the X chromosome. There is an equal probability of the fertilization of the ovum with the sperm carrying either X or Y chromosome. In case of ovum fertilized with the sperm carrying X chromosome, the zygote developed into female, and the fertilization of ovum with Y chromosome carrying sperm result into the male offspring. Thus, it is evident that it is the genetic makeup of the sperm that is determine the sex of the child. It is also evident that in such a pregnancy, there is always 50% of the probability of either of a male or a female child. It is unfortunate that our society women are blamed for the giving birth to the female children and have been ostracized and ill-treated because of his false notion. How is the sex determination mechanism different in the birds? Is the sperm or the egg responsible for the sex of the chicks? Mutation. Mutation is the phenomena which result in the alternation of the DNA sequences and consequently result in the changes in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomenon that leads to variations in DNA. As you will learn in chapter 6, one DNA helix runs continuously from one end of the other in each chromatid in highly supercoiled form, and therefore losses and gain of the segment of DNA result in the alternation in chromosomes. Since genes are known as the located of the chromosomes, alternation in the chromosome result in the abnormalities, aberrations. Chromosomal aberrations are commonly observed in the cancer cells. In addition to the above, mutation also arise due to the changes in the single base pair of DNA. This is known as the point mutation. A classical example of such a mutation is sickle cell anemia. Deletion and the insertion of the base of pair of DNA causes the frame shift mutation. The mechanism of the mutation is beyond the scope of this discussion at this level. However, there are many chemical and physical factors that induce mutation. There are referred to as mutagens. UV radiations can cause the mutation in organism. It is the mutagen. Genetic disorders, pedigree analysis. The idea that disorders the inherited had been prevailing in the human society since long. This was based on the heritability of certain characteristic features in families. After the discovery of the Mendel work, the practice of the analyzing inheritance pattern of the trait in human being began. Since it is evident that the control crosses that can be performed in pea plant and some other organism are not possible in case of the human being. Study of the family history about the inheritance of a particular trait provides an alternative, such as analysis of the trait in the several of the generations of the family is called pedigree analysis. In the pedigree analysis, there is the inheritance of the particular trait is represented in the family tree over the generations. In human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality of disease. Some of the important standard symbols used in the pedigree analysis have been shown in figure. As you have studied in this chapter, each and every feature in any organism is controlled by one or the other gene located on the DNA present in the chromosome. DNA is the carrier of genetic information. It is hence transmitted from one generation to another generation without any change or alteration. However, the changes or alteration do takes place occasionally. Such an alternation of the changes in the genetic material is referred to as the mutation. A number of the disorders in human beings have been found to be associated with the inheritance of changed or altered gene or chromosomes. Mendelian disorder. Broadly, genetic disorder may be grouped into two categories, Mendelian disorder and chromosomal disorder. Mendelian disorders are mainly determined by the alternation of the mutation in the single gene. This disorder is transmitted to the offspring on the same line as we have studied in the principle of inheritance. The pattern of the inheritance of such Mendelian disorder can be traced in the family by the pedigree analysis. Most common of the prevalent Mendelian disorder are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such Mendelian disorder may be dominant or recessive. By pedigree analysis, one can be easily understand where the trait is the question is dominant or recessive. Similarly, the trait may be also have a link to the sex chromosome. In case of hemophilia, it is evident that this X-linked recessive trait shown pedigree is shown for the dominant and recessive trait. Discuss with your teacher and design pedigree for the characteristics link both the autosomes and sex chromosomes. Hemophilia. This sex-linked recessive disease which show its transmission from the unaffected carrier females to the some of the female progeny has been widely studied in the disease. A single protein that is a part of the cascade of the protein involved in the clotting of the blood is affected. Due to this, in an affected individual is a simple cut will result in non-stop bleeding. The heterozygous female for the hemophilia may transmit the disease to sons. The probability of the female becoming a hemophilic is extremely rare because the mother of a such female has at least two carrier and the father should be hemophilic. The family pedigree of the Queen Victoria shows the number of the hemophilic descendees as was the carrier to the disease. Sickle cell anemia. This is the autosome-linked recessive trait that can be transmitted from parents to the offspring when both the parents are carrier for the gene. The disease is controlled by the single pair of the allele HBA and HBS. Out of three possible genotypes, only homozygous individuals show the disease phenotype heterozygous, and individual appears apparently unaffected by the carrier of the disease, and there is the 50% of probability of transmission of the mutant gene to the progeny. Thus, to exhibit the sickle cell trait, the defect is caused by the substitution of glutamic acid by valine. at the sixth position of the beta globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein result due to the single base of substitution at the sixth codon of the beta globin gene, GAG to GUG. The mutant hemoglobin molecule undergo in the polymerization under low oxygen tension, causing the changes in the shape of the RBCs from biconvex disc to elongated sickle-like structure. Phenylketonuria. This inborn error of the metabolism is also inherited as the autosomal recessive trait. Such affected individual lacks of enzymes that convert the amino acid phenylalanine into tyrosine. As a result of phenylalanine is accumulated, the converted into phenylpyruvic acid and the other derivatives. Accumulation of these in the brain result in the mental retardation. These are also exerted through the urine because of its poor absorption of kidney. Chromosomal disorders. The chromosomal disorder, on the other hand, are caused due to the absence and excess of the abnormal arrangement of one or more chromosomes. Failure of the segregation of chromatid during the cell division cycle result in the gain and loss of the chromosomes, called aneuploidy. For example, Down syndrome result in the gain of the extra copy of the chromosome of 21. Similarly, Turner syndrome result due to the loss of X chromosome in the human females. Generations of the family is called pedigree analysis. In the pedigree analysis, there is the inheritance of the particular trait is represented in the family tree over the generations. In human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality of disease. Some of the important standard symbols used in the pedigree analysis have been shown in figure. As you have studied in this chapter, each and every feature in any organism is controlled by one or the other gene located on the DNA present in the chromosome. DNA is the carrier of genetic information. It is hence transmitted from one generation to another generation without any change or alteration. However, the changes or alteration do takes place occasionally. Such an alternation of the changes in the genetic material is referred to as the mutation. A number of the disorders in human beings have been found to be associated with the inheritance of changed or altered gene or chromosomes. Mendelian disorder. Broadly, genetic disorder may be grouped into two categories, Mendelian disorder and chromosomal disorder. Mendelian disorders are mainly determined by the alternation of the mutation in the single gene. This disorder is transmitted to the offspring on the same line as we have studied in the principle of inheritance. The pattern of the inheritance of such Mendelian disorder can be traced in the family by the pedigree analysis. Most common of the prevalent Mendelian disorder are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such Mendelian disorder may be dominant or recessive. By pedigree analysis, one can be easily understand where the trait is the question is dominant or recessive. Similarly, the trait may be also have a link to the sex chromosome. In case of hemophilia, it is evident that this X-linked recessive trait shown pedigree is shown for the dominant and recessive trait. Discuss with your teacher and design pedigree for the characteristics link both the autosomes and sex chromosomes. Hemophilia. This sex-linked recessive disease which show its transmission from the unaffected carrier females to the some of the female progeny has been widely studied in the disease. A single protein that is a part of the cascade of the protein involved in the clotting of the blood is affected. Due to this, in an affected individual is a simple cut will result in non-stop bleeding. The heterozygous female for the hemophilia may transmit the disease to sons. The probability of the female becoming a hemophilic is extremely rare because the mother of a such female has at least two carriers and the father should be hemophilic. The family pedigree of the Queen Victoria shows the number of the hemophilic descentees as was the carrier to the disease. Sickle cell anemia. This is the autosome-linked recessive trait that can be transmitted from parents to the offspring when both the parents are carrier for the gene. The disease is controlled by the single pair of the allele HBA and HBS. Out of three possible genotypes, only homozygous individuals show the disease phenotype heterozygous, and individual appears apparently unaffected by the carrier of the disease, and there is the 50% of probability of transmission of the mutant gene to the progeny. Thus, to exhibit the sickle cell trait, the defect is caused by the substitution of glutamic acid by valine at the sixth position of the beta globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein result due to the single base of substitution at the sixth codon of the beta globin gene, GAG to GUG. The mutant hemoglobin molecule undergo in the polymerization under low oxygen tension, causing the changes in the shape of the RBCs from biconvex disc to elongated sickle-like structure. Phenylketonuria. This inborn error of the metabolism is also inherited as the autosomal recessive trait. Such affected individual lacks of enzymes that convert the amino acid phenylalanine into tyrosine. As a result of phenylalanine is accumulated, the converted into phenylpyruvic acid and the other derivatives. Accumulation of these in the brain result in the mental retardation. These are also exerted through the urine because of its poor absorption of kidney. Chromosomal disorders. The chromosomal disorder, on the other hand, are caused due to the absence and excess of the abnormal arrangement of one or more chromosomes. Failure of the segregation of chromatid during the cell division cycle result in the gain and loss of the chromosomes, called aneuploidy. For example, Down syndrome result in the gain of the extra copy of the chromosome of 21. Similarly, Turner syndrome result due to the loss of X chromosome in the human females. Failure of the cytokinesis after the telophase stage is the cell division. Result in the increases in the whole set of the chromosome in an organism, and this phenomenon is known as polyploidy. This condition is often seen in the plants. The total number of the chromosome in a normal human cell is 46, 23 pairs. Out of these, 22 pairs are autosome and one is the sex chromosome. Sometimes, though rarely, either the additional copy of the chromosome may include in the individual or individual may lack one pair of the chromosome. These situations are known as trisomy or monosomy of the chromosome, respectively. Such a situation leads to the very serious consequences in the individual. Down syndrome, Turner syndrome, Klinefelter syndrome are the common examples of the chromosomal disorder. Down syndromes. The cause of these genetic disorder in the presence of an additional copy of the chromosome number 21, trisomy on 21. This disorder was first described by the Langdon Downs in 1866. The affected individual is short, stunted, and small round head, furrowed tongue, and partially open mouth. Palm is broad with characteristic palm grace. Physical, psychomotor, and the mental development is retarded. Klinefelter syndrome. This genetic disorder is also caused due to the presence of the additional copy of X chromosome result into the karyotype of 47 XXY. Such an individual has overall masculine development. However, the feminine development of breast or gynecomastia is also expressed figure. Such individuals are sterile. Turner syndrome. Such a disorder in the caused due to the absence of the one X chromosomes and 45 with XO. Such females are sterile as ovaries are rudimentary besides other features including the lack of the other secondary sexual characters.